Good afternoon, everyone. Welcome and thank you all for joining us here today at the third Herbal Research Webinar Series 2025. It is my pleasure to invite our guest speaker today, Ms. Supaporn Intatham, to share with us a talk on anti-diabetic activities of medicinal plant recipes. Thank you very much, Ms. Supaporn, for taking the time today to share with us your discoveries. As usual, before we begin, we would like to remind all our participants to keep their microphones and videos switched off during the talk. You may switch them on again during the question and answer session. Without further ado, may I please invite our Herbal Research Group Lead, Dr. Nurul Aini, to give us a brief introduction about Ms. Supaporn. The screen is yours now, Dr. Nur.
Thank you very much, Dr. May, who is also the person behind organizing all this. Welcome all to the third Herbal webinar series for this year. As it's July already, but we're on to our third. Hopefully, we'll have more in the coming months. The webinar series titled today will be delivered by Ms. Supaporn Intatham, who is a research assistant at the Kanker Research Center for Food and Herbal Product Trial and Development from the Faculty of Medicine, Chiang Mai University, Chiang Mai, Thailand. Her current research focuses on evaluating the anti-diabetic activity of medicinal plant recipes in high-fat diet and streptozotocin-induced diabetic rats to obtain reasonable evidence before early feasible testing in human clinical trial. The floor is yours now, Ms. Supaporn, to deliver the webinar series titled Antidiabetic Activity of Medicinal Plant Recipe. And thank you from the organizer of Herbal Research Group for agreeing to share with us your finding on this webinar series. The screen is yours now, Ms. Supaporn. Thank you.
Okay. Thank you. Let me share my slide first. Okay, can you see my slide?
Yes, it's on perfectly.
Okay. Good morning, good afternoon, everyone. My name is Supaporn Intatham, and as you know, I am a research assistant at CFR at Faculty of Medicine, Chiang Mai University from Chiang Mai, Thailand. And today, I'm pleased to present my research on the anti-diabetic activity of the medicinal plant recipe. And this study was carried out into the 6 main steps. The first one, we did the plant selections and extractions. And then in the second step, we did an in vitro evaluations of the pharmacological activity of the plants. And after that, we summarize of the pharmacological activity of the plants. And then the first step, we preparations of the medicinal plant recipe. And next, we move on to the fifth step, which is the in vitro evaluations of pharmacological activity of medicinal plant recipe. And the last step, we focusing on the in vivo evaluations of anti-diabetic activity of medicinal plant recipe. And now let's take a closer look at each step in the study. The first one is the plant selection and extractions. We conducted a research across the several databases to identify the medicinal plants with reported the hypoglycemic activity in both human or in experimental animal. And from this, we selected a total of 7 plants, since some of the plants have known the active compound listed in the Thai Herbal Pharmacopoeia. So we proceeded to compare the different sources of the raw material to determine which one is contained the highest concentrations of the active compound. So the 7 selected plants are as follows. The first one is Panchakhan. Panchakhan was obtained from 2 sources. Lamphun and Chiang Rai, the Panchakan from Chiang Rai, from Lamphun sample were dry at the Faculty of Pharmacy, Chiang Mai University. And for the Chiang Rai sample were dyed at the different locations. The first one is the Faculty of Pharmacy, Chiang Mai University, and the second one is the Chiang Mai Royal Agricultural Research Center. And the second plant is Chiang La. Chiang Da we obtained from Chiang Mai, and it was identified into different leaf, the narrow reef and the broad reef. And both of this type of Chiang Ra were dry at the Faculty of Pharmacy, Chiang Mai University. And the third one is Obshoy. Obshoy was obtained from the Herbal Medicinal shop in Chiang Mai. And the another four plants is Baimon, Tamlung, and Marakinok, which were obtained from the Chiang Mai Wild King. We get this sample from Nan province, and all of these plants were dry at the Faculty of Pharmacy, Chiang Mai University. And these medicinal plants were extracted using the two different methods, the water extractions and the external extractions. For the water extraction, the plant including the Panchakan from Lamphun, Chiang Rai 1 and 2, Chiang Da for both of leaf type, and Obshơi, Baimon, Tamlung, and Marakinok. And for the ethanol extract, it consists of the Panchakan from Chiang Rai 1, Chiang Da, Baimon, Tamlung, Marakinok, and King. And now we move on to the second step. The second step is the in vitro evaluations of pharmacological activity of the plants. And before moving on to this step, I would like to briefly discuss about the pathophysiology of the diabetes and the mechanism of actions of the antidiabetic drug. And this figure shows what is known as the vicious cycle of the hyperglycemia, which play an important role in the blockades of the type 2 diabetes. An unhealthy lifestyle and metabolic dysfunctions can lead to increase the triglyceride and the fatty acid in blood, and the excess lipid accumulations in non-adipose tissue can disrupt the insulin signaling and cause the insulin resistance, especially in the liver. And this leads to the increased glucose productions and reduce the glucose uptake and rising the blood glucose and also the blood insulin levels. And elevated insulin promote the further lipid accumulations and worsening the insulin resistance. And over time, the high glucose and the lipid level damage the pancreatic beta cell, which impairing the insulin secretions and further increase the blood glucose level. And this image illustrates the role of the oxidative stress in the development of the type 2 diabetes. It occurs due to the low antioxidant level and increase the productions of the reactive oxygen species and caused by the metabolic overload, inflammations, and the stress from the high insulin demand. And oxidative stress then can contribute the insulin resistance and also cause the pancreatic cell damage and further worsening the type 2 diabetic conditions. And so to manage the type 2 diabetes effectively, both blood sugar control and also reductions of the oxidative stress is an essential. So the one approach to treating the type 2 diabetes is by inhibiting the enzyme alpha amylase and the alpha glucosidase, which help to control the postprandial blood glucose by slowing the carbohydrate digestion in the small intestines. The acarbose is the example of this drug that works through this mechanism. And the other drug is the metformin. Metformin helps improve the insulin resistance in the liver by reducing the fat accumulations. The metformin can increase the AMP level and then activate the AMPK and then inhibit the ACC1 and 2. And this can lowering the fatty acid synthesis and decrease the glucose productions and ultimately lowering the blood sugar level. So therefore, in this study, we focus on evaluating both the anti-diabetic activity and the hypoglycemic activity of the plant extract. In the anti-diabetic activity, we include the phytochemical screening on the total phenolic and the flavonoid contents, as well as the radical scavenging activity. And in the hypoglycemic activity, involve the alpha amylase and alpha glucosidase inhibitory activity, also the cytotoxicity assay. And finally, is a glucose uptake stimulation activity. And now I will explain the experimental method, also the result for each of these study. In conducting the phytochemical screening on the total phenolic and flavonoid contents, we use the following Shokutel assay to determine the total phenolic contents, and the method is that the plants were mixed with the following Shokutel reagent and the sodium hydroxide, and then incubated for 3 minutes. After that, the absorbance were measured by using the micropipette leader. And for determining the total of flavonoid content, we use the aluminum chloride calorimetric assay. So the plants was mixed with the sodium nitrite and incubated for 6 minutes. After that, the 10% aluminum chloride was added and incubated for further 6 minutes. And then the sodium hydroxide and distilled water were added later. And finally, the absorbance were measured by using the micropipette leader. And here is the result. The results show that the highest total phenolic and flavonoid contents were observed in the water extract of Obshey, Baimon, and Pachakan Lampun respectively. And among the ethanol extract, King, Tamlung, and Baimon show the highest level of the total phenolic and flavonoid content respectively. And next, in the evaluations of the radical scavenging activity, we carry out using the two assays, the DPPH assay and the ABTH assay. In the DPPH assay, the DPPH solution were mixed with the plant extract and then incubated in the darkness for 30 minutes. And finally, the absorbance were measured. And for the ABTH assay, the plant extract were mixed with the ABTS solution and then incubated for 6 minutes, and the absorbance was determined. And this present findings show the positive relationship between the high total phenolic and flavonoid content and the antioxidant activity. The water extract of Obshey, Baimon, and Panchakan show the high radical scavenging activity towards the DPPH and ABTH radical. And this result show that the ethanol extract of King, Tamlung, and Baimon exhibited the maximum radical scavenging activity obtained in the DPPH and ABTH assay. And based on the result mentioned earlier, we can summarize the plant with high phenolic and flavonoid contents, as well as the strong radical scavenging activity as follows. For the water extract, it consists of the Panchakan, Rampun, Obshey, Baimon, and Tamlung. And for the ethanol extract, we have the Panchakan from Chiang Rai, Baimon, Tamlung, Chiang Da, and King. And these selected plants were therefore chosen for further testing for their hypoglycemic activity. And next, we evaluated the hypoglycemic activity of each plant. The first assay focusing on the amylase inhibitory activity, and the second one is the alpha glucosidase inhibitory activity. The procedure for the alpha amylase inhibitory activity assay is as follows. The alpha amylase solution was mixed with the plant extract and then incubated for 10 minutes. After that, the CpNG3 as the substrate was added to each well and incubated for 10 minutes. And the third step, the reactions were terminated by additions of the sodium carbonate and the absorbent were measured. And for the alpha-glucosidase inhibitory activity, the reaction mixture consisting of the alpha-glucosidase solutions were pre-mixed with the plant extract and then incubated for 10 minutes. After that, the PNG were added and incubate for further 20 minutes. And the reactions were terminated by adding of the sodium carbonate and the absorbent were measured. Here is the result of the assay. In the group of water extract, you can see that only just Tamlung had the alpha amylase inhibitor, while the Obchoy and Baimon exhibited the inhibitory activity against the alpha glucosidase. And among the ethanol extract treated, King has the highest inhibitions of the alpha amylase amylase activity, followed by Chiangda and Baimont. And the inhibitory activity of King was only higher than that of the standard acarbose, whereas the ethanol extract that have the alpha glucosidase inhibitory activity range from the most to activity to the least activity. Baimon, Panchakhan, Chiang Rai 1, Chiangda, Tamlung, and the inhibitory activity of these plants was higher than the standard acarbose. And this slide summarizes the result of the alpha amylase and the alpha glucosidase inhibitory activity. Alpha amylase inhibitions was observed from the water extract of Tamlung and the ethanol extract of Baimon, Chiangda, and King. And for the alpha glucosidase inhibitory activity, the effective plant, including the water extract of the Obchoy, Baimon, and the ethanol extract of Pachakhan Chiang Rai 1, Baimon, Tamlung, and Chiangda. We then carry out the cytotoxicity assessments of all plants using the HRB assay. The Hep2 cell were seeded in the 96 well plate and incubated for 24 minutes. After that, the plant extract was added and incubated for 24 and 48 hours. And after that, the 20% TCA were added and incubated overnight. After that, the plate was washed with the tap water, and then the HRB solution was added to each well and incubated for 1 hour. And then rinse the plate with the 1% acetic acid to remove unbound dye. And then the tris-based solution was added to the plate. And the optical density in the microplate were measured by using the microplate leader. And from the result, the extract were cytotoxic to the Hep2 cell in the different concentrations. The group of the water extract that exhibited the high IC20 and IC50 were considered relatively safe. From the most to the least are Panchakhan from 3 sources and the Baimon and Tamlung, respectively. And among the water extract, you can see that the Obchoy demonstrated the most potent cytotoxicity extract based on its lowest IC20 and IC50 value. Therefore, this extract was considered unsuitable for further development into a recipe. In the group of the ethanol extract, the high IC20 and IC50 value were found in Baimon, Tamlung, and Panchakhan respectively, indicating the low cytotoxicity. And following the cytotoxicity assay, we proceeded to investigate the glucose uptake stimulation activity. The Hep2 cells were seated in the 96-well plate and then incubated for 24 hours. After that, the medium was changed to an insulin-containing serum-free medium, and the sample was further incubated for 24 hours. And then the serum-free medium contained insulin were replaced with different concentrations of the plant extract and incubated for 24 hours. Which the concentrations of the plant extract used in this study, it didn't exceed the IC20 value as demonstrated in the previous slide as the cytotoxicity study. And at the end of the incubation period, the medium was removed and placed in the new plate, and then the glucose concentrations was determined by using the glucose assay kit, and the optical density were measured by using a micropage reader. And the results show that the 3 water extract with the strongest effect on the stimulations of the glucose uptake in the Hep2 cell was Baimon, Tamlung, and Panchakan from Rampur. And this slide presents the effect of the ethanol extract on the glucose uptake, and notably, the high glucose uptake stimulations were observed in King, Tamlung, and Panchakan from Chiang Rai 1. This slide highlights the plant extract with the most pronounced glucose uptake stimulation effect. For the water extract, this including Panchakan from Rampur, Baimon, and Tamlung. For the ethanol extract, it is the Panchakan Chiang Rai 1, Tamlung, and King. So, based on the result from the in vitro assay, we now arrive at the third step, which is the summary of the pharmacological activity of the plant extract. The potent candidates for the medicinal pen recipe were proposed. In the water extract group, Panchakan from Rampur, Baimon, and Tamlung show the most promise. And in the ethanol extract group, the Panchakan, Chiang Rai, Baimon, and King are the promising extract. And however, after we considering the mechanism of actions and the cytotoxicity result and supporting the evidence from the literature, the 4 herbal extracts were selected as the component for the final formulations. This include the water extract of Panchakan from Rampur and Tamlung, and the ethanol extract of Baimon and King. And you can see that most of these plants demonstrated the high pharmacological activity with no observed cytotoxicity. And the fourth step is the preparations of the medicinal pen recipe. The medicinal pen recipe can be prepared either by the research estimations or with the aid of the software to help calculate the optimal ratio in the recipe, such as the studies program. But in this study, we estimated the recipe ratio based on the effective doses of the plant extract observed in the in vitro study, combined with the literature review of effective doses in the animal study. We then calculated the ratio between the dose used in animal model and those used in vitro, and used this ratio to guide the formulations process. As a result, the 3 herbal recipes were developed, and each consisting of 3 different plant extracts as shown in this table. And since this study primarily focusing on the hypoglycemic activity of the Panchakan, so it was included in our recipe. And we developed the 3 medicinal pen recipe as follows. Recipe 1, including the water extract from Panchakan, ethanol extract from Baimon, and ethanol extract from King. And for the second recipe, consisting of the water extract from Panchakhan, ethanol extract from Baimon, and water extract from Tamlung. And the last one, the recipe 3, is composed of water extract from Panchakhan, ethanol extract from King, and the water extract from Tamlung. And the specific proportions of each extract in this recipe are shown as in this table. And we calculated the proportions of the plant extract in each recipe so that the total amount it would equal to 500 milligram. This this standard diet. These standard diets allow for more meaningful comparison between the medicinal plant recipe and the standard log, which is the metformin, both in terms of preparing the tablet and the equal dosage, evaluating the cost differences. And moreover, is able for fair comparison of the efficacy of the medicinal plants and the metformin at the same dosage in the further study in humans. And after completing the herbal formulations, the next step, step 5, involves the in vitro evaluations of pharmacological activity of the medicinal plant recipe. We tested all 3 medicinal plant recipes for their in vivo inhibitory activity of alpha amylase and alpha glucosidase, as well as the glucose uptake stimulation activity, which the experimental method used were the same as those applied in the single herb extract study. And from the alpha amylase inhibitory activity, we found that none of the medicinal plant recipe show a significant inhibitory activity against this enzyme. Therefore, we conclude that the targeting this enzyme may be not suitable for the further development, and the data for the alpha amylase inhibitory activity are not presented here. But however, the result of the alpha glucosidase inhibitory assay showed that both recipe 1 and recipe 2 exhibited a dose-dependent inhibitions of this enzyme. And among them, the recipe 2 demonstrated the slightly stronger inhibitory activity than the recipe 1 at all test concentrations. And in contrast, the recipe 3 showed no inhibitory effect on this alpha glucosidase. Therefore, the data are not presented here. And in the glucose uptake assay, we observed that in the insulin-resistant cell had a low glucose uptake compared to the control group, and all 3 medicinal plants were able to increase the glucose uptake more than the control. And recipe 3 showed the strongest effect at the concentrations of 100 microgram per milliliter, and while the other concentration, the recipe 2 showed the best activity. And the mechanism of action of the medicinal plant recipe are summarized in this table. Recipe 1 and 2 demonstrated both the alpha glucosidase inhibitory activity, while the recipe 3 show only the glucose uptake inhibitory activity. So based on this finding, the recipe 1 and recipe 2 were selected for further investigations of their anti-diabetic activity in the high-fat diet and streptozotocin-induced diabetic rats. The final step, the 6th step, is the in vivo evaluations of anti-diabetic activity of medicinal plant recipe. This is the plans of the study diagram. The male Vista rats were fasted for 12 hours, and the baseline fasting blood glucose were measured using the glucometer before starting either the normal diet and the high-fat diet. After that, the normal group were given the normal diet. and the diabetic group were given the high-fat diet. And all of these two groups will receive the diet for 8 weeks. And after that, the normal diet group were injected with the seated buffer, while the high-fat diet will receive the 30 milligram per kilogram streptozotocin or HTZ for induce the diabetes. And 1 week after receiving the HTZ, the fasting blood glucose was measured using the glucometer before starting of the treatment. And in the diabetic group, the rat with the blood glucose level between 250 to 400 milligram per deciliter was selected for the study. And the normal rat were divided further into 3 groups, and for the diabetic rats were divided into 8 groups. For the 3 groups of normal rat is the normal rat receiving distilled water as group 1 for the control group, and the second group and the third group is the normal rat that received the recipe 1 and recipe 2 at a dose of 200 milligram per kilogram. And for the diabetic group, it were divided into 8 groups. The group 1 is the diabetic rat received distilled water as a control group, and the second group is the diabetic rat received metformin at a reference group. And the other 3 groups is the diabetic rat received recipe 1 at a dose of 50, 100, and 200 milligram per kilogram. And other last 3 groups received the recipe 2 at a 3 dose at the same as recipe 1. And the rats were treated with this recipe for 4 weeks. And throughout the study, the blood glucose level were measured weekly in all groups, and body weights were recorded, and the food and water intake also monitored. Additionally, an oral glucose tolerance test or OGTT were performed both before and after treatment. And at the end of the treatment period, the rats were euthanized and the bloods were collected for analysis of metabolic parameter, lipid profile, hematological parameter, and the pancreas was removed, weighed, and preserved for the histopathological examination. So I will now present some of the initial findings as the study is still in progress. And let's begin with the effect of the medicinal pen recipe on normal rats. In this experiment, we monitor changes in the metabolic parameter both before and after treatment, as you can see here. And this table shows the body weight of the normal rat before and after treatment with the medicinal pen recipe. We found that the normal rat treated with R2 or recipe 2 show the less body weight gain than those in the control and the R1 group. This may be due to tamlung in the recipe 2, which can inhibit the fat accumulations, while king in R1 may enhance the digestion and nutrition absorptions and leading to the greater weight gain. And this graph is the present the pancreas weight per body weight ratio. In the normal rat treated with the R1 and R2 show no significant difference in the pancreatic beta cell pancreatic to the body weight ratio compared to the normal control group. And next, the food intake of the normal rat before and after treatment with the medicinal pen recipe is shown in this table. The normal rat treated with both recipe 1 and recipe 2 or R1 and R2 show reduced food intake. This may be attributed to panchakhan, which is a component of both recipe, which is known to activate the AMP, activated protein kinase, and enhancing the cellular energy utilizations and suppress the appetite under the normal conditions. And in addition, the normal rat treated with R1 consume less food than those treated with R2. This may be due to the presence of the king in R1, which enhance the digestion and nutrition absorption and lead to an earlier sense of fullness. And for the water intake, the normal rat that received the R1 drank less water than those receiving R2. However, the water intake in R2 treated rat did not decrease significantly, is suggesting that the weight loss is what likely due to the other effects, such as the reduced fat accumulation, but not from the water intake. And this table shows the effect of the medicinal pen recipe on the fasting blood glucose level of normal rats. Normal rats treated with R1 and R2 show no change in the fasting blood glucose level, indicating that both of these medicinal recipe has no effect on the fasting blood glucose in the normal rat. And this graph illustrates the glucose area under the curve, or AUC value measured during the OGTT test. Before and after treatment, the AUC value in R1 and R2 normal rat were similar to the control, indicating that the normal pancreatic functions and no glucose lowering effect under the normal condition. And this table presents the lipid profile of the normal rat before and after treatment with the medicinal pen recipe. After treatment, the recipe 1 significantly reduced the triglyceride or TG, while the recipe 2 significantly lowered the total cholesterol or TC. Despite these changes, all values remain within the reference range. And this table shows the hematological parameter of the normal rat. After treatment, all groups exhibited a significantly increase in MCV compared to the before treatment. However, this value remained close to the reference range. And the table on this page shows the effect of the medicinal pen recipe on the different white blood cell count. Or normal rat group show a decrease in the white blood cell count after receiving the medicinal pen recipe compared to the baseline as the before treatment. However, this value remain within the reference range. And these are the current study on the effect of the medicinal plant recipe in the normal rat. And now I will present the result of the effect of the medicinal recipe on the diabetic rat. In the diabetic rat, you will see that at week 9, our diabetic rat show the weight loss after HTZ injections, and R1 and R2 treatment lead to the weight gain, but not to pre-diabetic level. And the weight gain was lower in the metformin or MF group and the R2 group when compared to the R1 group. And this study, we study the pancreatic weight to the body weight ratio of the diabetic rat. And we found that the diabetic rat treated with metformin and recipe 1 or R1 at a dose of 100 and 200 milligram per kilogram show an increased pancreatic to body weight ratio compared to the diabetic control. This may be related to the metformin known effect in reducing the inflammation and oxidative stress in the pancreas, while the R1 effect may stem from the plant component, which can help to protect the beta cell by reducing the oxidative damage. And this table show the food intake of the diabetic rat before and after treatment with the medicinal pen recipe. And at week 9, all groups show reduced food intake follow the STZ injections. And after treatment, the diabetic rat received metformin continue to show decreased food intake. But in contrast, those that treated with R1 and R2 show increased the food intake and approaching the pre-diabetic level as shown in week 8. And notably, the group that received the R1 rat can consume more food than those received the R2. And this slide presents the water intake of the diabetic rat. After week 9, oral group show increased water consumptions after STZ injections, and treatment with both metformin R1 and R2 lead to a reductions in the water intake. This table highlights the key findings on the effect of the medicinal pen recipe on the fasting blood glucose in the diabetic rat. You can see that at week 9, all groups show the elevated fasting blood glucose level followed by the STZ injections, and a reduction in a glucose level was observed in MF group starting at week 12 and week 13. In addition, the diabetic rat treated with R1 at the dose of 100 mg per kilogram and R2 at the dose of 50 mg per kilogram show a decreased glucose level beginning from week 11 to 13. And these findings are consistent with the early result, which show that the medicinal plant recipe help normalize feeding behavior and also reduce the water intake, which both reflecting the improve of the blood glucose level. And in this slide, show the AUC of glucose during the OGTT in diabetic rats. No significant difference were observed between the group before and after treatment. This may be due to the 24-hour gap after we giving the treatment before testing the OGTT, allowing all of the test substance already clearance. And however, the reductions in the fasting blood glucose shown in the previous slide may be attributed to the other mechanism of the medicinal pen recipe, such as the antioxidant activity or enhanced glucose uptake and inhibitions of the carbohydrate digesting enzyme, such as the alpha glucosidase, as demonstrated in the in vitro study. And this table shows the lipid profile of the diabetic rat before and after treatment with the medicinal pen recipe. After treatment, all of the diabetic group show a reductions in the triglyceride or TG level compared to the baseline as a before treatment, approaching the reference range. This decrease may be due to physiological adaptations follow the diabetes inductions, particularly increased use of the fat for energy in response to the glucose deficiency, rather than the direct effect of the test substance. The effect of the medicinal pen recipe on the hematological parameter are shown in this table. After treatment, some of the diabetic group show increased hemoglobin or HB and hematocrit or HCT compared to the baseline before treatment. However, this value remain within the reference range. And the final result is the differential white blood cell count of the different rats after treatment with medicinal pen recipe. All diabetic groups show a decrease in white blood cell count compared to the baseline before treatment, though the value remained within the different range. It is possible that 4-week treatment durations was not sufficient to produce a clear change in the hematological parameter, as the blood system may require more time to respond to the test substance. And these are the current findings on the anti-diabetic activity of the medicinal pen recipe in high-fat diet and streptozotocin in diabetic rats. And please note that this is not a complete database, as the study is still ongoing. And further evaluations, including the plasma insulin level, glycosylated hemoglobin level evaluations, and also the histopathological analysis of the pancreas. And this study are still underway still ongoing, and these or results are underway to support the final interpretations. And nevertheless, the preliminary study suggests data that I shown here suggest the promising potential for this medicinal plant recipe to be developed into the herbal product for blood glucose control and further evaluating in the human study. And before I conclude, I would like to take a moment to acknowledge the key contributor to this research. First, I would like to thank you to Associated for Professor Dr. Siwaboon Siliratawong as our project leader and consultant for his guidance and expertise. And second, Assistant Professor Dr. Suni Chansakal has provided the herbal raw material and extract essential to our study. And finally, the Dr. Virakit Teshawarodisakun carried out the in vitro experiment and supported the data generations and analysis. And I hope this presentation has provided a clear overview of the research and its significance. Thank you for your attention.
Thank you very much, Ms. Supaporn, for your very interesting talk. That's a lot of work that has been done on all these plants. So, before we move on to is there anything you want to add before we move on to the group photo session, Dr. Nurul?
No, well done. I'm so impressed with the presentation, how you simplified everything and highlight, and then you work on. It's a stepwise process whereby your group have worked on based on the evidence that there is. I have few questions in mind, but before that, let us do group photo, which we usually do before everyone go, Miss Supaporn.
Yes.
Thank you very much. Well done.
If we could have everyone turn on their cameras, hopefully everyone has gotten the background from the email that we hope most of our audience are willing to have on their background for a more cohesive look. We'll give everyone a few minutes to turn on their cameras. Okay. Hi. I'm still getting ready. Okay. So on the count of 3, I'll take the picture on the first page. 1, 2, 3. Okay. All right. Give me a minute. Okay, now on the second page, on the count of 3, 1, 2, 3. Okay, very nice. Right, thank you everybody. So now we can move on to the question and answer session, which I think a lot of us are looking forward to. Do we have any questions from the floor? I think we have one in the chat. Oh, okay, that's just Dr. Nurul sharing the background. Thank you, Dr. Nurul. Dr. Nurul, I think your mic is switched off.
Okay,
There we
yeah.
go.
Yeah. Question from the audience,
Or yeah.
participants, please?
Anyone wish to ask any questions? They can turn on their mics, or if you would prefer, you could type the questions down in the chat box and we
Yeah.
will read it out for you. So while waiting, maybe Dr. Nurul, you can address some of your questions to Ms. Supaporn first.
Right, yes. Oh, hold on. I was playing around with my. Okay, Miss Pampon, congratulations and well done for the for sharing this. In fact, I'm very happy because one of the herbs are the herbs we're working on, the Morris alba, and it's nice that you also put the local name next to your presentation. So that's nice to know. And it's Morris alba is one of the is in one of the recipe one and recipe two, medicinal plant recipe. Yeah. So it's also nice. Like normally people work on one plant and just test it on one plant, whereas your group are working on various plants. First you screen off, and then you narrow down to 3 plants. Why do you choose 3 plant instead of 5 plant or 4 plant? Is this based on the ethnobotanical use, or these are based on uh, yeah, if you can explain why do you choose just 3 plant in one recipe? Why not 2? Why not 4? Why not 5 plant?
From this, I'm not sure, but because the first thing that the method how to we use to select the plant to into the recipe is based on the our project leader, the Dr.
I see.
Supaporn. Yes,
Your
and
associate professor, right?
yes,
Okay.
and what I see it does most of not just only this study but also the another study I have seen that they select just 3 plants. I
Just
think
3 plant.
was
Okay.
yeah one of the factor that they use just 3 plants. I think because they are also like also uh want want to when we prepare the recipe, we have to worry about like the uh how to say that is the synergistic,
Yes,
right?
synergistic effect.
Yeah,
Yes.
yes,
I
yes.
I also noted in some research group
Mm-hmm.
they only work in a very small plant because of
Yes.
this. But sometimes they work only just 2 plant. But I'm this is my first time seeing 3 plant
Mm-hmm.
in one recipe. So maybe this will also have some synergistic
Yes.
effect to each
Yes.
other.
Yes.
So but how do you choose that the ratio? I know you put ratio
Oh, yes.
vitro, but it was not clear. 1:3:2 or something like that.
Let
But
me see.
overall, it's all 500 milligram, right?
Mm-hmm.
But how do you
Um,
choose? I know
in
you,
this slide.
yes,
Oh,
I know you
sorry.
put in vitro and in vivo and also literature review, but how do you choose? This is it. This is the recipe. So I wonder also whether it's linked with the ethnobotanical use, what the local use, or it has nothing to do with what the local use. These are based on the experimental in vitro result.
Uh, you can see my slide.
Yeah, yes.
Okay. Actually, as I talk with, my uh project leader, he said that actually it is not the like the standard method for
Mm,
to mix of the plant into the recipe, but this method that I show you today is from his method, It's not
okay,
like the standard method. Yeah. It is
okay.
what he think is that he want to know what is the effective dose in the in vitro. In vitro is mean the single cell, right? Because we test
Mm,
just in the cell lines. And he want to know what is the effective dose in the animal or
Animal
in
control.
in the body, the system, not only one cell. So he create this two table, the effective dose in the research experiments or in vivo or in single cell, compare with the effective dose in the literature review, which come from the animal study. So finally he divided the dose from the in vivo to in vitro, like for example, like 22.5 and divided by 0.05, and you will
You
get
get
this.
okay.
Yes. So and in this number is mean that how much of the effective dose in the animal compared to the how much effective dose from the in vitro. So we
I
uh,
see.
yes, compare all of these plants and we get all of this. And also, so we after we choose like four plant, so we as I told you that he want to create the recipe as three of plants in the recipe. So we random all of three plants in different recipes. So we get three recipe and how of how the all of this number come from, right? We just it's like the thought for the mathematic. So we sum of all of this, sum all of this number, and we just change to the sum of all of this number
The 250.
to the
I see.
500.
So much.
Yeah,
Yeah.
yeah.
Okay.
This
So
is
that's
the
just a
in the direct way. Yes.
guesstimation. Okay,
Uh-huh.
nice to know. Nice to know. Yeah, because as I said, this is my first time seeing people presenting in the form of recipe.
Uh-huh.
Normally, with TCM, traditional Chinese medicine, normally it's linked to what the local use.
Oh,
So these
yes.
are more ethnobotanical use, whereas
Mm-hmm.
come up with this recipe because of this literature review and also research evidence or research
Mm-hmm.
experimental value, right?
Yes.
Okay,
Yes.
good to know.
Mm-hmm.
Oh, interesting, interesting. And also, perhaps I wanted to know, like, the plant that you work on, because there's so many extract you need to come up. Do you take them in one in the same area location, all the plant, or it's in various part? You know, Thailand is very big. Like, can it be from Bangkok? I don't know. Like, maybe it's northern is different from
Yes.
Bangkok, right? Thailand is big.
Uh-huh.
Now it's not too big.
Yes.
Do you take from one plant or one trees or various location? The one that you
Uh,
work.
in this part is working by the Dr. Suni from the Faculty of Pharmacy, that they will work for selected the plants and also extract the plants for us. But as I know, all of the plant is not all in each plant. They cannot they do not collect from the same locations. And
Uh-huh.
as I show you, that's one of them, like Panchakan, is also from Lamphun and
Lampung.
also from
Okay.
Chiang Rai. Yes.
Okay, that's the
And
name of
another
the
one,
area.
yes, and the king also from Nan. But I want to know, I will tell you that all of this province is also in the northern of Thailand.
Northern of
Yeah, maybe
Thailand.
it's
Okay.
yeah, it's around in the northern of Thailand, because it's not far from, like, our location in Chiang Mai, right? Yeah, it's not
Right.
easy to find that plant. Mhm.
Okay. Do people actually? Oh, okay. It is question from Zoe. One question from the participant. Hi, thank you very much for the talk. Would like to know, with the measurements of the rat's pancreatic weight, body weight ratio, does a higher ratio means that the treatment shows benefit in the rats? It's
Uh,
yeah.
Yeah, the measurements of the rat uh, pancreatic weight to body weight ratio is calculated from, we measure the pancreas weight from the rat, right? And divided by the body weight of each rat. Actually, this method is used when all of the rat not in the same weight, the different rat, the different weight. But we want to normalize, the ratio. So we do the pancreatic body weight divided by the uh, pancreatic weight divided by the body weight. Yeah, and in this, the higher ratio mean that the treatment show benefit in the rat. I think that is correct. Based on like another study, they finding, for example, that some plants, if that plant can increase the pancreatic per body weight ratio, it means that pancreatic after it was damaged by the STZ injection, right? The STZ injection can result in the decrease of the pancreatic weight. So if you give the plant to the rat, and that plant can cause to increase the pancreatic weight, it is maybe you can assume that that plant maybe have any mechanism that maybe activate the pancreatic to more divided to create more pancreatic cell, resulting in the more pancreatic weight. Yeah. And more of the pancreatic waste, as you know, the pancreatic is the cells of, they have the beta cell that can produce the insulin for control the blood glucose. If you have increased the pancreatic weight, maybe you can better to control like the blood glucose in the rat.
Just another question. Thank you very much, Fazul. There's another question from Muhammad Abdul Rashid. Do you have histology data, especially pancreas data?
Oh, for now, we do not have that because the study is still on progressing. Yes, now we just preserve the beta, but before waiting for sending them for evaluating the histological
Yeah.
pathology.
Otherwise, you
Yes.
would have shown on your slide,
Yes.
right?
Yes.
Thank you very much. What other question I wanted to know? I think these are for my master student. Where are you guys? Do you want to ask questions? I think they always like to know whether when preparing for the experiment, do you freshly prepare your herbal extract or you can keep it for some time and then do. I notice you also work on animal and there's 8 group and all that. There is a lot of extract preparation there. Do you prepare it fresh or you can conserve or preserve it in a fridge or things
Oh.
like that?
Yeah, for the preparations of the raw material and the plant extract is what performed by the Dr. Suni from the Faculty of Pharmacy.
Oh,
After they extract the plant, they send it to us at the Faculty
ah,
of Medicine,
okay.
so that we
See?
keep it in the fridge like -20, for waiting for the animal is ready and testing is in the animal. Yes.
Okay.
Mhm.
From Semarang University, thank you very much. Nice presentation, comprehensive research from Rohadi from Semarang University. Thank you for joining us. Any other questions?
I do have a few questions
Yeah,
actually. On
please.
the note of extraction, may I just know what kind of extraction method was used for this entire project? I know there's the water extract and the ethanol extract. So what was the method that was used by Dr. Suni to prepare the extracts, if you have any idea?
Oh yeah, actually, as I show, it's just like the overall of the extract. Like for example, like the water extract, we have a lot of plants, but actually they have like a specific of the detail method of each plant that is not exactly the same. Yes, but I didn't show here today. But I think it's also based on both of the literature review that they do, and also based on the Thai herbal pharmacopoeia that they use, because when we extract, right, we have to have like the standard, how to say, the bioactive compound that have to control the quality of the both raw material and the plant extract, right?
The
Yeah.
So it's based on the
Yes.
optimized
Yes.
extraction method.
Yes.
Wow, that is impressive.
Mm.
Okay. So, on
In
another
Thailand, you have the herbal pharmacopoeia as well, right?
Yes, Thai
Yes,
herbal
Thai
pharmacopoeia
herbal
for the
pharmacopoeia.
standards reference. Yes.
So you follow based on that. I wonder
Yes.
if these selected medicinal plant are also being used traditionally. I mean, yes, maybe ginger definitely,
Mm-hmm.
but the rest of the plant, like Morasalba, you also use them?
Yeah. Yeah.
Okay.
Also use them in Thai.
Oh,
Yes.
so
Mm-hmm.
that's why there's a Thai name of it,
Yes. Yes.
right? So these medicinal plant selection are based on what's being used
Mm-hmm.
by your local people, right?
Yes.
Okay, so because yeah, because there's no rationale on that in your presentation.
Mm-hmm.
one question, Doctor.
Uh, Other question again? Have you checked the toxicity before inducing anti-diabetic dosage? Toxicity.
Toxicity. For this study, we didn't do the toxicity before inducing the anti-diabetic dose, but I think they just looking at the literature review, maybe we can know like the primary result that some of plant that have the component of the recipe in the literature review, we already know the dose of the toxicity and animal, and we compare that to our research that used the plants in the recipe. Yeah. And I think after do this anti-diabetic activity and before we using it or study it in the clinical trial in human, we also need these informations about the toxicity of the herbal recipe. Yeah, before perform it in the human clinical trial because it's just very important. Mhm.
It's not shown, yeah?
Yes.
Thanks. Right. So you wish to pursue clinical trial of this recipe to human in the later stage of this study?
Yes,
Oh,
because our center we also have both to
you're the
do
center, yeah.
yes, also do as
Mm.
our center, yeah.
Oh, yeah. We used to send our student, pharmacy student, to Chiang Mai
Oh,
University.
really?
Yeah, two of our students, but I
Mm.
think it's not Faculty of Pharmacy. It's
Oh,
maybe biochemistry. Our first cohort
oh,
of pharmacy student, we send
mm-hmm.
our student for discovery just for 3 months.
Oh,
So it'll be nice to explore or reignite our relationship on
yes.
that. Yeah, we hopefully can send student in the
Uh-huh.
future. So we'll touch base with your supervisor or
Mm-hmm.
yeah, or at the center
Yeah.
in the
Yes.
future. Yeah.
Yes.
Okay, that's great to know.
Yes.
Yeah, the labs look very impressive from your website. Yeah, more
They're
than
the
18
center, right?
pharmacies. Yes.
Is this center like for the whole of Thailand or just Chiang Mai
I
area?
think from the whole of Thailand.
From
Yeah.
the whole of Thailand.
Yes.
So it's Chiang
Mm-hmm.
Mai is the center, not
Just you mean how many uh the
center of
center do you have? Oh,
herbal
just only here in Chiang
only
Mai.
there.
Yeah,
Oh,
but
no
I
one.
mean that we also work with the another organizations around the Thailand.
Mm,
Yeah.
mm. Do you also collaborate with international organization like
For
send your sample or etc., or formulate it? No,
no
just
for now no,
for now
just
it's
just from the Thailand because our center just around I'm not sure maybe 2 or 3 years for just have this center, but
oh
it is
being formed
yeah,
2 3
yeah,
all right
yes,
after COVID
yes, mm-hmm,
after COVID then
yes,
All right. It's new.
Yeah, it's still
No
new.
wonder. Okay, okay.
And we
Because
are like, oh, very excited that we have the
oh,
contact from you from like
Oh,
yeah,
different
yeah,
country. Yeah.
yeah. We'll
we'll be
think
in,
of
yeah.
collaborating with your
Yeah.
center because
Mm-hmm.
it sounds very good. You work together, yeah?
And
Hmm.
how can I ask how do you find like our informations?
Or Dr. May to answer.
Oh.
Because we had some feedback from our audience. They were interested
Uh-huh.
on anti-diabetic studies. So I had a
Mm-hmm.
search on the literature and I came across the paper from Dr. Sirawun. So yeah,
Mm.
from then I started contacting and
Uh-huh.
one thing led to the other and now you're presenting for us today.
Yeah, and doctors you would, oh, it's your work. You can to do.
Yeah, you should be proud of your work. That's a very impressive thing you have done there.
Mhm.
Okay, so Dr. Rohadi has to leave from Semarang University. He has to leave, so I'll share the attendance form and feedback form as well, just for anyone who is in a rush to leave. But we can carry on with our Q&A session if anyone else has any questions to ask. Talking about
Yeah.
toxicity, actually, I recall on one of your slides you showed the results for cytotoxicity, isn't it? For
Mhm.
the plants. So I didn't quite um catch the conclusion from that slide. Do you mind showing that slide again?
Oh,
Just
yes.
on the IC, I think it's IC 20 and IC 50.
Yeah.
Uh-huh. This one is the
Ah,
result of the water extract.
okay. Also, some of the IC 50 even went up to 3,000, close to 4,000. Ooh.
Yeah.
So those are considered um, low in activity, right? Because the IC 50 is very high.
No, it means that this plant is really safe, because they have
Ah,
IC50. It means that these concentrations can inhibit the life of the cell
right.
50%,
Yes,
Yeah, so if you have uh if you have to use
so much.
like for example, like Panchakan Chiang Rai 2, right?
Yeah.
To kill the cell, 50% of the cell, you have to use the concentrations at 300, 3,000 and around 900 microgram per
Yes.
milliliter. It means that you have to use like
A lot before
a
it
lot.
counts the
Yeah,
cells. I
yeah.
see, I
But
see.
yes, but compare with the
Hmm.
obchoy is quite like cytotoxicity because
Yeah.
you use just only like 77.
Oh.
Yeah.
That's a bit concerning because cinnamon, this is the cinnamon that we
Yes, yes, this is the
use in the
cinnamon.
kitchen. I see.
Yeah,
So we cannot
yes.
overdose ourselves with cinnamon then. Oh, it harms the liver. I see.
Yes.
These are done on the liver cells, isn't it?
It tastes used no in the oh yeah, yeah, liver
Liver.
cancer cell, HT2. Yes.
What about Chengda? What plant is that actually? That one has quite a low
Shem.
IC20 as well,
Nairobi.
Yes.
58. Oh,
Shengda is I think it's like our local plant, in northern Thailand.
I
It's
see.
known as no to
Mm.
lowering the sugar level.
I
Yeah,
see.
so we also focusing on this, but no, it's not conclude in the recipe.
No, the
Mm,
Morris Alba is. Why do you? It's
yeah.
interesting. You put narrow leaves, broad leaf. Is that like
Mm,
mature and young?
No, I also asked like the Dr. Siwabun and Dr. Suni, they said it is the same species of the plant, but it just um the what we see to them is the narrow or the broad. I'm not sure. Maybe I'm not sure, maybe different location or any effect that cause them have the different appearance. Yeah, you know.
Possibly.
But
I think
but
So
Mama
but
different.
Salva has
yeah.
different shaped leaves as well.
Yeah. But I
Mhm.
said, but he said, it is also the same species. Okay.
Okay.
Maybe
Yeah,
different location. I'm not sure.
that's so interesting. Thank you
Mhm.
very much for sharing that with us. I think that's all from me for questions. So, yeah, do we have any other questions from the audience? Anyone who wish to speak into their mics? If not, we might try to conclude. I think we are going a bit over time now. 20 minutes past. Sorry to take up your time. Okay. So I believe that should be it since no one is responding. Perhaps I'll share the slide on the QR code so it's more convenient for everyone to scan to access the forms. Thank you
Uh,
very much, Ms. Supaporn.
yeah, thank you very much on behalf of our UBD Herbal Research team. We would like to thank you, Ms. Supaporn Intatham, for presenting a very nice slides and comprehensive, very easy on the eyes presentation of your medicinal plant recipe, how you derive from that. I think we've learned a lot, and it's very interesting because some of the herb, as I said, also overlap with what we're doing, and that is a very exciting news as well. Like for us, like, and unfortunately, I think July is it's a like a term break for us, so not a lot of students joining. Otherwise, we have normally we have double the attendance. Yeah, Dr. May. So,
Yes.
but anyway, we have recorded your presentation. We can share with those researchers who are not joining us this afternoon. But yeah, we look forward to collaborating with you, perhaps because you're the center of research on herbal plants. So we could be sending students in the near future to do discovery year, whereby we can all expenses are on us. We can send them for 3 months to help also learn some of the technique you're doing there under your doctor's supervision. Thank you very much. We'll be in touch for that.
Thank you. Thank you so
Thank you. Thank you
much.
so
Thank you
much,
very much
everyone.
too.
And thank you for those who joined us.
Yes, thank you for all our participants.
Yeah.
Some are very regular participants.
It's always the same name.
Yes,
We
good to
like to
see
hear your feedback.
everyone. Yes, please
Well,
share with us.
Thank you again. If you have like any questions or want to like collaboration, you also can contact me or Dr. Sibaboon. Yeah.
Great.
sure.
Thank you so
Yeah.
much, Mr. Popcorn.
Yes. And hope to like yeah, collaboration with you again in the next future.
Thank you so much.
Thank you very much.
Okay. I think we still have
Page with
some
an
more
icon.
people. Yes.
Thank you, Dr. May.
Thank you so much, Dr. Nurul, for sparing your time during your leave. I will discuss with you again later on the other method the other pressing method. Okay, I think we still
Okay,
have, no, the
I'll
rest
leave
should still, yeah.
first.
Okay,
Yeah.
sure, doctor. I'll just leave this on for a while. Hey, is everyone else still doing the form? I will leave the form on for another 2 minutes for everyone to complete the attendance and feedback. Then I will end the Zoom session. Thank you everyone again for joining.